Hello, welcome to the lecture series for MLT 120, Hematology and Hemostasis. We'll be learning exactly what hematology is, and we'll also be learning about parts of the cell. What is hematology? Hematology is the study of blood and blood disorders, and a hematologist or a hemopathologist is a highly qualified, trained healthcare provider who specializes in the study of diseases of the blood and the blood components. Things that a hematologist or hemopathologist may study include the blood itself, bone marrow or the bone marrow cells, hematological tests that help diagnose anemia, infection, hemophilia, blood clotting disorders, and leukemia. In the average adult, we have about 5 liters of blood in total in the body. The overall function of the blood, there's several of them. First off, the blood helps transport oxygen from the lungs to the tissues. Then the blood also helps clear tissues of CO2. Blood helps transport glucose, proteins, and lipids throughout the body. And finally, blood helps move waste to the liver and the kidneys from different areas of the body. When we're talking about the different types of cells that are contained within the blood, there's 3 different types of cells that are in your blood. First, we have the red blood cells, which carry the oxygen. These are also referred to as erythrocytes. So if you hear erythrocytes, we're referring to red blood cells. Next up, we have white blood cells. These are helpful in fighting off things like infections, and it helps give immunity to the body. We call these leukocytes. So if you hear leukocytes, we're referring to our white blood cells. And then finally, we have our platelets. These are what helps our blood to clot and helps stop bleeding of the body. And these are also referred to as thrombocytes. So again, if you hear thrombocytes, we're referring to platelets. Let's take just a second to look at the image that's on this slide. And this is a blood vessel, a representation of a blood vessel. This red tube, this is your blood vessel. And all these are the different cells and components that are contained within your blood vessel, and this is your blood, the cells that make up your blood. So these little red discs that look kind of like donuts, these are representing your red blood cells. These larger ones that are kind of clear and have different shapes inside of them, these are different types of white blood cells. Okay, so we'll be learning later on the different types of white blood cells. These smaller little spiky looking figures, these are your platelets. So there's a platelet and there's a platelet. This liquid looking material that's in between your cells, that's your plasma. And we haven't talked about plasma yet. Now that we have seen plasma in the previous slide, let's talk about exactly what plasma is. Plasma is the liquid portion of the blood. Plasma transports your blood cells around in the body. So your red blood cells, white blood cells, and platelets are all suspended within the plasma or the liquid part of the blood. Plasma also transports constituents such as glucose, electrolytes, hormones, proteins, and lipids around the body through the blood vessels. Now that you know what plasma is, let's talk about the difference between plasma and serum. Sometimes you'll hear those terms used interchangeably, but there's actually a difference between plasma and serum. We typically refer to the liquid part of the blood when it's inside the vessels of the body as plasma. But however, in lab testing, we also refer to the liquid part of the blood as serum sometimes. And there's a particular situation or something that happens in the lab that causes it to be serum versus plasma. Plasma is when it's in your body and the blood is moving through the vessels and the liquid is not clotted. When it's not clotted. Okay, so remember that. So let's take a second and look at the image that's on the right hand side of this slide. Let's think about if we take a patient's blood, let's take a sample from a patient, and we collect it in a tube that's got an additive that doesn't allow the blood to clot. So if it doesn't clot, it's not clotted, then the liquid portion is going to be plasma. Okay, so that's what we have here in this tube. The tube has not been allowed to clot, or the blood has not been allowed to clot. So if you divide it up into parts, what's in the tube, you've got red blood cells, you're going to have your white blood cells and platelets, and the liquid portion is going to be plasma because the tube has not been allowed to clot. And in your plasma, you can have things like different nutrients, water, proteins. There's all kinds of things that can be contained within that plasma. So we've been talking mainly about plasma. So let's talk about what serum is. Serum, once the blood clots, it will contain cells and other constituents that pull away from the liquid part of the blood. This liquid part of the blood is now serum. So to get serum, you've got to allow the blood to clot. So when we draw our blood into a plain tube with no additives, remember on the previous slide we had an additive in the tube that didn't allow the blood to clot. So you still have plasma, but in this situation we've got a plain tube, so it's naturally going to clot over time. And when it clots, the liquid part of the blood is now going to be referred to as serum. And you can see a comparison. The purple top tube has an additive that doesn't allow the blood to clot, like we saw in the previous slide. So you still have plasma as the liquid portion. But over here is a red top tube, which is also referred to as a plain tube, and it does not have that additive, so it allows the blood to clot. So you end up with this clot in the bottom of your tube, so your red blood cells clot together, so the liquid portion of the blood is now referred to as serum. So why would we want serum versus plasma? Because some lab tests require a serum specimen versus a plasma specimen. So thus we must understand the difference between a plasma specimen and a serum specimen in the laboratory setting. Okay, let's say you're working in a laboratory and the phlebotomy team or the nursing team brings you a bunch of samples in tubes. What they're going to bring you is whole blood. And this is before it's been separated out in either plasma or serum. So the way we usually separate it out is to use a centrifuge, and this is an example of a centrifuge. We put the whole blood in the tube into a centrifuge and we spin it at so many RPMs in order to separate out the components. And we usually spin blood at about 3,500 RPMs for about 3 to 5 minutes, depending on what your laboratory's protocol is. But you're going to be putting a tube of whole blood in, and depending on what type of tube the blood was collected in and what type of additives was in that tube is dependent on whether you're going to end up with plasma or serum. So if you have an anticoagulant additive in your tube, that's not going to allow your blood to clot. So you're going to end up with a plasma sample after it's spun down in the centrifuge. If you have a plain tube and it does not have an anticoagulant, it's going to allow the blood to clot. And you do have to allow a little bit of time in between the blood being collected from the patient and it being spun down. You have to allow 5 to 10 minutes for the blood to clot before it's spun, and then you'll end up with serum as your liquid portion of the blood. So just remember, no clot, plasma, clot, serum. So we've already talked about this a little bit, but if a lab test specimen, the requirement is plasma, we must find a way to draw that blood into a tube that allows that specimen not to clot. Okay, we don't want to clot if we want plasma. So to achieve this, we must draw it into a tube that has an additive that's called an anticoagulant. So no clotting. So we draw the blood into a tube containing anticoagulant, so the blood will never clot due to the effect of the anticoagulant. If we don't spin it down, though, usually spin it down in the centrifuge and separate them out. But if we just leave it sitting out on the counter, eventually the red blood cells in the tube will settle to the bottom of the tube and the liquid portion will be on the top. And that liquid portion, of course, will be plasma because there was never a clot that was allowed to form. Okay, just to make sure that we're good and clear on the difference between plasma and serum, let's ask ourselves a couple of questions. Number one, if you draw a patient's blood into a plain tube, there's no anticoagulant additive, what will the liquid portion of the blood be? That would be your serum because you have no additive, so the blood is allowed to clot. So you end up with serum. Number two, you draw a patient's blood into a tube that contains an anticoagulant. What will be the liquid portion of the blood called? In that case, we've got plasma because we do not have a clot, or a clot was not allowed to form. When you've been to the doctor before or to the hospital, you may have noticed that when you've had blood drawn, there's different colors of caps of tubes. This is because in the lab we use different tubes that contain different types of anticoagulants depending on the tests that are being performed. And you can see just an example of some of the different types of tubes and the different colors of tubes that can be used or utilized in the lab in the image below. For this unit, we will learn a few types of tubes and anticoagulants in order to understand as we get further into the course and as we discuss different tests and required specimens. And how we're going to learn these different tubes is by the color of the tube cap, the anticoagulant that the tube contains, how the anticoagulant stops the blood from clotting, finally, the area of the lab where that tube is most commonly used. So let's dive into the different types of anticoagulants. Let's start out with the most common type of anticoagulant used. It is the EDTA tube, which is also referred to the lavender top tube or the purple top tube. It prevents blood from clotting by binding calcium. Binding calcium is also referred to as chelating calcium. Calcium must be present in our blood plasma in order for the blood to clot. If we bind up all the calcium in the specimen, the blood cannot clot. So we use this tube primarily in the hematology section of the lab. So it's used for hematology testing usually. Again, it's a purple top tube, and you have the examples over here on the far right hand side. You can have larger volume tubes, little short tubes, or you can have these little micro tubes that are usually used for finger sticks. Next up, we have our anticoagulant tubes that are sodium citrate. The additive is sodium citrate. The color is usually a light blue or a light blue colored cap. It prevents blood from clotting by binding. Again, chelating is another word for binding calcium. So it uses the same mechanism as our EDTA tubes, and this tube is primarily used for coagulation testing or clotting testing. Coagulation is also known as clotting. Next up is our green top tube. This is another commonly used anticoagulant tube. And in the green top, the anticoagulant is heparin. It prevents blood from clotting by inhibiting thrombin formation. Thrombin is a blood constituent that helps the blood to form a clot. If we stop it from forming, the blood in the specimen will not or cannot clot. And this tube is used primarily for chemistry testing. And you can see some examples of the green top tube there on the right hand side. The last anticoagulant we're going to talk about is the gray top tube. It contains the anticoagulant sodium oxalate. It prevents blood from clotting by also binding calcium. And it's usually used for some miscellaneous lab tests such as glucose, blood alcohol, and even lactic acid. And you can see an example of the gray top tube there on your right hand side. So all the tubes we were just talking about that contain anticoagulants, you would end up with a specimen of plasma. But now let's look at some plain tubes. And your plain tubes, you're going to end up with a specimen of serum. So usually our plain top tubes are usually red top tubes, but they also could be maybe orange top or maybe gold top or even what referred to as a tiger top. And yes, if these tubes are all referred to as plain tubes, they do not contain anticoagulants. So these tubes are going to allow your blood to clot, and therefore you're going to end up with your serum as your sample. So most plain tops do not have an additive, but some will contain a additive that actually accelerates your blood to clot. So those additives are called a clot activator. Remember earlier on in the slide I said you have to wait. If you immediately draw a tube from a patient before you spin it down, you have to wait 5 or 10 minutes before you spin that sample down to allow the blood to clot. Well, sometimes these plain tubes can have a clot activator in them to help speed up the clotting process of the blood, so you'll get that specimen of serum a little bit quicker. So to sum up this slide, your plain tubes, you'll end up with the specimen of serum. Here's a really nice chart of the tubes that we've been over. It shows the tube color, the additive for each tube, the function of the additive, and the common area where that tube is used in the laboratory. I would encourage you to pause the video at this time and to really look over and try to commit to memory these tubes, because these are the tubes that are most commonly used in the laboratory. Here's a trivia question to review the information that we have just learned. Which of the following anticoagulants below does not bind calcium? And we've got EDTA, heparin, sodium citrate, and sodium oxalate as our options. So if you want to pause the video at this time and try to answer before you look at the next slide that has the answer, you can do that. Of course, the answer to our trivia question is heparin, and that's because heparin inhibits thrombin formation to prevent the blood in the tube from clotting. And this will end up, or you'll end up with plasma as the liquid portion of the blood in a heparin tube. This concludes part 1 of our first hematology lecture. I hope you've enjoyed learning about what hematology is and about the different anticoagulants. And I will see you over in part 2 where we'll be talking about the cell.